Hey everyone, here we are. It's Monday, August 3rd, and hopefully everyone is on track to get their exams in by 5 o'clock this afternoon. And then we're actually in the home stretch. I'm planning to get through the rest of the chapters as outlined in the syllabus. Even though we've fallen a couple of chapters behind, I think we can still get there. I posted a minor revision of the syllabus last week when I realized that I wouldn't get through chapter 12. Well, at least without having everyone need to watch extended lectures. I always try to limit them to the 75-minute class period, even if a few of them have been a little bit shorter, like probably this one will be. Now, in today's lecture, I'm going to talk about the final stages of memory consolidation. And actually, as I say that, I hesitate, because we still need to consider memory reconsolidation. But that's coming up in chapter 14. So for today, we're going to look at ongoing memory maintenance, and we will again be focusing somewhat, or well, actually pretty much exclusively, on the role of PKM zeta. Remember the kinase protein or memory molecule that induces a cycle of self-replication at potentiated spines to provide ongoing management of those spines to overcome the impacts of molecular turnover. We've seen the PKM zeta manages or maintains the integrity of the system by phosphorylating AMPA receptors and keeping up the elevated level of insertion and diminishing the removal of these receptors from the spine heads. So we'll look at that in terms of memory consolidation or maintenance today. And the other topic we'll cover, a much less studied topic, is, let me see. What was it I was going to say? Oh, yeah. The neurobiology of forgetting. Haha. So, in the previous chapter, chapter 11, we were talking about the neurobiology of memory consolidation. Now, today we're going to move beyond memory consolidation and talk about memory maintenance. During consolidation, we saw that LTP is very vulnerable to disruption, and the period of consolidation in tetanized sections of hippocampus seemed to be on the order of, well, about 2 to 4 hours. The vulnerable period for memory in the in vivo context, in living animals, in behavioral experiments, and in people appears to be somewhat longer. And we typically define consolidation of memory, the period of greater lability of memory, as lasting up to about 24 hours. Beyond that period, of course, is the maintenance period, in which the spine has been remodeled, the AMPA receptors are in place, and the spine's not so dependent upon synthesis of proteins from top mRNAs and upon the genomic cascade. Of course, this is the period when the spine seems to become most dependent upon maintenance of elevated synaptic activity by the actions of protein kinase M zeta. Now, the experiment depicted on this slide is actually quite straightforward, but there are 3 parts to it. So I'll show you the first part here, behavioral test, and then there's going to be another behavioral experiment on the next slide. And then the one after that will have some biological concomitants to support the data from the behavioral experiments. So let's look first at this behavioral experiment. In this experiment, Janine Quapis and colleagues in Fred Helmstedter's lab conducted a contextual fear conditioning experiment in rats. Now, this was a standard fear conditioning experiment, as we've seen so many times before. And if you look at the timeline, you can see that the rats were trained to form the association between the foot shock and the context at the beginning of the experiment. Then after either 1 day or 7 days, the rats were injected with the protein kinase M-zeta inhibitor, zeta inhibitory peptide, or ZIP, into the basolateral amygdala. And you can actually see the microinjection sites clustered all in or around the basolateral amygdala in the figure at the left. And you can see lots of these sites here contralaterally across these regions that show the basolateral amygdala. Now, for the control rats that were injected with saline 1 day after training and then tested 2 hours later, they seem to have a robust memory of the contextual fear conditioning as they froze for about 80% of the test session. For the rats that were injected with ZIP into the basolateral amygdala 1 day after training and then tested 2 hours later, they seem to have a very reduced memory of the contextual fear association, as they actually only froze for about 35% of the test session. And then the other cohort of rats that was injected and tested 7 days after training, we can see that for the control rats injected with saline, that 7 days after training and tested 2 hours later after that injection, they still have a robust but slightly reduced memory of the contextual fear association, as they froze for about 60% of the session. And for the rats injected with ZIP, 7 days after training. So hypothetically, they had the memory enduring like the control animals for those 7 days. But now they're injected with ZIP 7 days after training and tested 2 hours later. They also have a very, very reduced memory of the contextual fear association, as they actually froze for only about 30% of the test session. So it seems that you can disrupt protein kinase M-zeta maintained memory with ZIP injected into the basolateral amygdala, either 1 day or 7 days after the conditioning, indicating that protein kinase M-zeta is an essential component of memory maintenance at both time points. Of course, this isn't a big surprise given everything that I've told you about protein kinase M-zeta so far, but up till now we've mostly looked at it just in terms of LTP in a section of hippocampus that isn't going to endure for a long period of time. These experiments are showing us that even after 7 days after incorporation of the memory, that the memory is vulnerable to disruption when you administer a protein kinase M-zeta inhibitor. Now, in the second phase of the experiment, the same groups of rats were tested 24 hours after the first test. Again, this is illustrated in the timeline. Of course, we've already seen that the rats were trained in the contextual fear conditioning and then injected with either saline or ZIP 1 or 7 days after the training. They were tested 2 hours after the injection, as I showed you on the previous slide, but then they were brought back again and tested 24 hours after that initial test, that initial injection test. Now what we can see is that the freezing was further reduced in the saline-treated animals, reflecting perhaps some forgetting and also perhaps the extinction influence of going through test number 1. However, you can clearly see that there's no recovery of the memory in the 24 hours after test number 1 in the animals treated with ZIP. They continue to show very little freezing when placed into the previously conditioned context. Now, this suggests that the ZIP was not having just some transient effect, temporarily disrupting either memory or performance in test number 1. It seems that once you just transiently or for a short while interrupt the function of protein kinase M-zeta with a treatment like ZIP, you are going to permanently impair the maintenance of that memory. Okay, but you might argue that perhaps the ZIP might still be present and continue affecting the performance of the ZIP-treated rats. Well, Quapis and colleagues, I guess they kind of anticipated that concern, and they have that covered too, as I'll show you on the next slide. Now, in this experiment, Quapis and colleagues showed that ZIP is actually completely eliminated by 24 hours post-injection. You know, come to think of it, I don't think I've described the ZIP drug in any detail, but it's simply a peptide that functions as a pseudo-substrate that binds to the catalytic region of PKM-zeta, almost like a regulatory unit, which we know that PKM-zeta doesn't have. But by binding to it, it inhibits its catalytic activity. So I'm sure that you can imagine that a peptide like ZIP could be rapidly degraded. And of course, a peptide could also be labeled for an immunohistochemistry experiment. So the figure on this slide shows the degradation of ZIP over 24 hours after injection. You can actually see the labeled ZIP at the tips of the injector cannulae in the brain sections at 0, 2, 8, 14, and 24 hours post-injection. And the graph shows that ZIP immunofluorescence was completely eliminated by 24 hours. So the bottom line is that the ZIP disrupted the memory when it interfered with PKM-zeta, with protein kinase M-zeta function acutely. The spines that carried the index of the memory relaxed back to their naive state, and appears that access to the memory was permanently lost. I also want you to notice that I described that the index of the memory was disrupted and access to the memory was lost. I didn't say the memory was disrupted by injections of a drug into the basolateral amygdala, and that's because the amygdala, and for that matter the hippocampus, do not store the complex memories. We know that the memories for a task like fear conditioning are encoded in all their complexity in distributed networks of many thousands or millions of synapses in the cortex. So I'm going to resist the temptation to go down the rabbit hole of what is the role of the amygdala and hippocampus in memory function. But we will be coming back to this topic in chapter 16. Meanwhile, just keep in mind these ideas, that the amygdala and hippocampus may encode a memory index to promote memory access, not the actual encoding of the entire memory itself. And there I've given away another punchline prematurely again. Okay, let's look at a completely different kind of behavioral experiment shown on this slide. Now, breaking away from our standard cued and contextual fear conditioning, and also outside of our regular focus on the amygdala and hippocampus. This one takes advantage of a very, very powerful form of learning known as conditioned taste aversion. In this experimental paradigm, a rat or a mouse is offered an opportunity to taste a new substance, a new pleasant taste. I've seen tastants like Kellogg's Blueberry Nutri-Grain bars, which I'm sure you could imagine is pretty exciting to a rat who's fed these grain pellets all the time. But the simplest to use, the most commonly used, and the easiest to measure is saccharin in the drinking water. Rodents love it. And all you have to do to measure this is to measure the starting and finishing volumes in the bottle, and thereby you can assess consumption. Okay, after allowing free access to a choice between water or saccharin solutions in a taste session, you can measure the volume of each and assess the preference for the saccharin. And then inject the animals with saline, shown on the left, or lithium chloride, shown on the right. The lithium chloride will induce nausea. And thereafter, the lithium-treated animals will very robustly avoid the saccharin-tainted water. This is a conditioned taste aversion. And of course, you can imagine that this powerful form of learning has a huge evolutionary significance. Because what if animals just ate and drank whatever was available, regardless of the consequences? Well, you can imagine that evolution would come to a screeching halt for those animals. Conditioned taste aversion is such a powerful paradigm that we see that it occurs in one trial learning, as opposed to other conditioning experiments where you have to expose the animal to the association between, let's say, the cue and the foot shock repeated times before they catch on to that association. Conditioned taste aversions are particularly powerful. They can be formulated in one trial. And I should also tell you that the insular cortex, and that's the part of the brain where gustatory cortex resides, that this area of cortex has been implicated in conditioned taste aversion. Because, well, if you lesion or pharmacologically block the insular cortex prior to training in conditioned taste aversion, then you block the acquisition of that conditioned taste aversion. And let's move on and look at an experiment with this paradigm. So Shima, Sacktor, and Dudai used microinjections of ZIP to study the role of protein kinase M-zeta in this powerful form of learning. In this first experiment, they offered the animals a choice between saccharin and plain water for their drinking water. And then they injected the rats with lithium chloride. Control rats were injected with saline, and throughout the experiment, the data from the control rats were used to calculate the aversion index for the lithium chloride treated rats. If the lithium chloride treated rats drank a lot less saccharin than the saline treated rats, then the aversion index is high. And if the lithium chloride treated rats drank just as much saccharin as the saline-treated rats, then the aversion index is low. In fact, the dashed line across the middle of the graph indicates equal consumption of saccharin and water. So in this experiment, after the saccharin exposure and the lithium chloride or saline treatment, then 3 days later, they injected ZIP or vehicle into the insular cortex. So you can follow the timeline at the bottom of the slide, and you can also see the location of rat insular cortex in the rhinal fissure in this diagram at the lower right. And for comparison, I also included a diagram showing the human insular cortex, where the insular cortex lies inside the lateral fissure. Okay, so in one cohort of ZIP or vehicle-treated rats, they waited 7 days before testing. And then they tested in 3 daily tests. In the other cohort, they actually waited a full month before testing in the 3 daily tests. Now, overall, you can see that the vehicle-treated animals had a decline in the aversion index over time across those 3 tests, indicating that there's some decline in the association or memory. However, there's still a robust conditioned taste aversion. The rats treated with ZIP, in comparison, have a very low aversion index, both for the rats treated with ZIP and tested after 1 week, and the rats treated with ZIP and tested after 1 month. By the second test, in both of these groups, they don't seem to show any preference for saccharin over water or water over saccharin. So it looks as if protein kinase M-zeta in the insular cortex is implicated in conditioned taste aversion, just as we saw that it's implicated in the amygdala in fear conditioning. Furthermore, disrupting protein kinase M-zeta for a brief period of time, and remember we saw that the ZIP is metabolized by 24 hours post-injection, but disrupting for just that brief period of time disrupted the memory, even when tested 1 month later. In a second experiment, Shima, Saktor, and Dudai injected the ZIP or vehicle 3 days, 7 days, or 25 days after the taste aversion conditioning. In the rats injected with ZIP after 3 or 7 days, they tested the conditioned taste aversion just 2 hours later. But in the rats injected after 25 days, they tested the conditioned taste aversion actually 24 full hours later. And you can see that under all these conditions, the rats that had the PKM-zeta inhibitor injected into their insular cortex were impaired in the conditioned taste aversion in comparison to the vehicle-treated controls. And so clearly, protein kinase M-zeta is implicated in the maintenance of access to this memory, and you can even disrupt it as late as 25 days after consolidation of the memory, and you see impacts upon the ongoing maintenance. Now let's have a look at the last of the 3 experiments that they reported in their 2007 science paper. In this one, They conducted the standard taste aversion conditioning with saccharin plus lithium chloride or saline and tested the animals 3 days later. You can see that all the animals have a robust aversion to the saccharin, a high aversion index. Then, 1 to 4 minutes later, they injected ZIP or vehicle and then tested the animals daily for the next 3 days. And then they took a pause, and then they tested again on days 12 and days 14 to 16. Obviously, the ZIP knocked out the conditioned taste aversion. It knocked out the memory for the association between the saccharin and the nausea. Now, the vehicle-treated animals show some decrease in the aversion over days 1 to 4, perhaps due to a decline in the memory or, again, an effective extinction, but they still have a robust aversion that even shows some spontaneous recovery after the no testing days when they were retested on days 12 through 14. Note that there is no recovery in the ZIP-treated animals. And even when the ZIP-treated animals were re-injected with lithium chloride on day 13, they showed no recovery of the conditioned taste aversion. The conditioned association is well and truly lost when protein kinase M-zeta function is just briefly inhibited after the first test. Four years later, Shima and Dudai were at it again with another science paper on protein kinase M-zeta. This time, they used a viral vector, or lentivirus in this case, to overexpress protein kinase M-zeta or to express a dominant negative construct of protein kinase M-zeta into the insular cortex. The image of a rat brain shows the location of expression of the vector tied with a green fluorescent protein sequence into the insular cortex bilaterally in the rat. Oh, and let's explain what a dominant negative construct is. It is, in this case, a gene construct encoding the protein kinase M-zeta protein, but it's mutated at a critical site in the protein, preventing its kinase function. So imagine if the cell is flooded with a construct that makes a non-functional protein kinase M-zeta, then it just might interfere by being trapped by activated spines and substituting for the functional protein kinase M-zeta. Except, of course, that it doesn't work. So the effective factory worker is replaced by its useless good-for-nothing cousin, and the whole factory is messed up. Okay, wow, that analogy was a real stretch. Sorry about that. Okay, now the Western blot and the graph over on the right side of the slide show that administration of the lentivirus encoding full-length protein kinase M-zeta produces over a 5-fold increase in protein kinase M-zeta protein. And of course, the control vector encoding just green fluorescent protein, or GFP, does not. Now, in this experiment, I think it's also important to note that when they wanted to show the potential enhancement of conditioned taste aversion by injecting a gene construct encoding a fully functional protein kinase M-zeta, They didn't max out the conditioned taste aversion in all animals by using a high concentration of lithium chloride. Of course, instead they used a low concentration and allowed the construct to work to boost up that aversion in the animal. And when they wanted to show potential impairment of conditioned taste aversion by injecting a mutant gene construct encoding a non-functional protein kinase M-zeta, they did actually use a high concentration of lithium chloride so they could effectively knock down the impact. So let's see what happened on the next slide. Let's look first at what happened when they injected the insular cortex with a dominant negative gene construct encoding a protein that we think should interfere with the effects of the endogenous functional protein kinase M-zeta. The timeline at the top shows that the animals were conditioned for taste aversion on day 1. And then after 5 undisturbed days, on day 7, they were injected with lentivirus containing either the dominant negative construct or just a gene encoding green fluorescent protein, a control vector. Then they waited for 6 days and then tested on days 14 and 15. And of course, you can see that the rats injected with the control virus, the green fluorescent protein, maintained their aversion, just as you would expect. But the rats injected with the gene construct encoding the dominant negative protein showed impaired aversion. They appeared to have lost the memory for the conditioned aversion, as the mutant protein is getting in the way of the function of normal protein kinase M-zeta. Once again, we have a demonstration that protein kinase M-zeta is essential for the ongoing maintenance of the conditioned taste aversion memory. And now, perhaps more exciting, let's see what happened when they actually overexpressed normal, functional protein kinase M-zeta into the insular cortex. In this case, they injected the viral constructs and then waited for 5 days so the virus could incorporate and start to robustly make protein kinase M-zeta before the taste aversion conditioning. And then you can see that the rats injected with the control green fluorescent protein vector have a mild conditioned taste aversion to the low dose of lithium chloride when tested on days 9 and 10. And in contrast, the protein kinase M-zeta overexpressing mice exhibit an enhanced, very robust protein kinase M-zeta after treatment with the low dose of lithium chloride. In this third part of the experiment, they first conditioned the rats on day 1, as opposed to injecting the vector on day 1. So note how this differs from the previous experiment. In the previous experiment, they injected the animals with the vector first, allowed it to incubate, and establish the overexpression of protein kinase M-zeta. So now they trained the rats first, waited for 5 days, and then injected them with the lentivirus and gene construct encoding the full-length, fully functional protein kinase M-zeta on day 7. And under these conditions, the protein kinase M-zeta treated rats still showed a robust enhancement of the condition taste aversion. So in the experiment on the previous slide, the availability of protein kinase M-zeta at the time of the learning enhanced the ongoing retention of the learning. And this slide shows that even after the learning had been established, I suppose as long as it's still robustly present, the addition of more protein kinase M-zeta still enhances the memory. It seems as if the excess protein kinase M-zeta can be targeted to the activated spines and still used post hoc to enhance the memory. This really is a startling discovery. Think about it. All the other chemical treatments that can enhance cognition are drugs that are administered in the consolidation period, like amphetamine and caffeine. And all those drugs produce only really small improvements in cognitive function, with lots of side effects, including, of course, addiction. And we also see with all of those drugs that higher doses impair learning. So what if we really could produce an intervention like PKM-zeta that promotes long-term memory and reduces forgetting? Couldn't that have profound implications for treatment of cognitive impairments in aging, for example? What would you think if we could inject you with a lentivirus into your hippocampus, and then you go off and study hard, using good study habits so you don't exhaust your supply of Krebs, of course, and then you can take advantage of your lentiviral overexpression of protein kinase M-zeta to never forget the material for your organic chemistry test. Or you could even do your studying and then show up for your cognitive booster injection of lentivirus. Wouldn't that be exciting? Okay, but before we get too excited about these possibilities, maybe we should consider a couple of things. Consideration number 1, age-associated memory decline may involve additional pathological processes like degeneration of neurons and deficient cardiovascular function and formation of masses that prevent normal function of neurons. Protein kinase M-zeta mRNA or DNA expressed through a lentiviral vector might not really be the silver bullet that would deal with all of those problems. And consideration number 2, not many of us are going to want to have holes drilled into our skulls so viral vectors can be micro-injected into our hippocampus just to do better on our orgo exam. And even if you did, there's no guarantee it would work. We previously saw promising effects with drugs like ampakines, but the initial excitement has dwindled as these drugs didn't quite live up to their promise, didn't live up to their hype. Also, we don't know what the long-term risks and side effects might be if you repeatedly injected your hippocampus with a viral vector. So yes, this is still science fiction, and there's nothing better for learning in your coursework than plenty of sleep and good study habits. Sorry. Okay, at the beginning of the lecture, I outlined that we were not only going to talk about protein kinase M-zeta and memory maintenance, but we were also going to talk a little bit about the neurobiology of forgetting. So let's move ahead and do that. Let's think about these cellular functions in a larger context. Human experience tells us that most events in daily life are actually forgotten. If I ask you about the circumstances surrounding your breakfast on June 17th, 2020, most, if not all of you, cannot reconstruct those circumstances. Okay, yes, there's some people who claim to have highly superior autobiographical memory, otherwise known as hyperthymesia or HSAM, HSAM. And perhaps the most well-known of these people is Marilu Henner, an actress from a 1970s sitcom named Taxi. Now, these reports are highly questionable, although some evidence indicates that the people who claim these abilities spend an inordinate amount of time focused upon their past, rehearsing and actually practicing these memory events. Nevertheless, for the rest of us, we remember important and emotionally salient events, sometimes through extremes of rehearsal, like studying for your Orgo exam, or sometimes because they are emotionally charged, like when your boyfriend left you, or when your little sister was born. Sorry, I hope I didn't bring anybody down with the former example or send you into paroxysms of mirth that would distract you from the rest of the lecture with the latter example. Okay, so unimportant and irrelevant associations are lost constantly throughout your daily life. Memory is enhanced when practiced, both through repetition of the experience or through repeated trials where one rehearses the experience over and over. And in addition, memory is enhanced when emotionally stimulating or when emotionally challenging. We have at least two theories to explain the selective storage of memories. One is interference theory, and one is active decay theory. Of course, these are psychological theories that emanate from and have their early origins in the field of cognitive psychology. However, like so many theories from the field of cognitive psychology, they're supported by evidence in the field of neuroscience, especially the theory of active decay. But before I get too far ahead, let's explore the psychological theories. Interference theory posits that new or additional experiences overwrite the encoding of recent events so that new experiences crowd out or interfere with the retrieval of less recent but not yet consolidated memories. In contrast, the active decay theory suggests that new memories, if not emotionally salient or practiced, simply degrade over time in the fairly short term. Let's examine the evidence. The best evidence we have is from some behavioral pharmacology experiments in rats. In these animals, tetanic stimulation was administered in vivo, stimulating the perforant path inputs to the dentate gyrus. The experiment was done in vivo so we can trace the potentiation across days rather than the few hours that we see in hippocampal sections. And so this in vivo stimulation produced LTP that was sustained across a few days and then declined. However, when the NMDA receptor antagonist CPP was administered, the LTP with the same stimulus parameters was sustained for 9 days or more. Let's think about that. The NMDA receptor is the principal source of synaptic influx of calcium in postsynaptic dendritic spines. If we block the function of the NMDA receptor, we can block the decay in synaptic potentiation. And so it seems that all we need for decline of synaptic potentiation is just the ongoing small influx of calcium through the NMDA receptors. And then the graph on the right shows that reference memory on the radial arm maze is preserved over time by administration of CPP. In this experiment, the number of reference memory errors went down with repeated trials. And then the animals were left undisturbed except for daily injections of CPP across 5 days. And at the end of 5 days, the test of reference memory in the radial arm maze revealed that the CPP-treated animals, shown with the closed circles, made fewer errors than did the animals treated with vehicle in the interim. Once again, it appears that just blocking the small amounts of calcium influx diminished the decay of the memory. Moreover, Hrabatova, and I'm sure I'm butchering that name, but Hrabatova and Saktor showed that if the synapses are stimulated with low-frequency, weak depolarizing pulses, the kind of parameters that would induce long-term depression, or LTD, then expression of conventional protein kinase C isoforms is increased. This appears to compete with expression of protein kinase M-zeta through an unknown mechanism to decrease the expression of protein kinase M-zeta. The figure at the top left shows immunoblots or Western blots for the protein kinase C isoforms. In all these isoforms, you can see that 3 Hz, 3 times per second stimulation increased the expression of conventional protein kinase C. And those data are also summarized in the graph at the bottom left. But then the graph at the right shows that protein kinase M-zeta and protein kinase C zeta lambda, the one that replaced protein kinase M-zeta in a previous experiment that we looked at a few chapters ago, these are both significantly reduced after the 3 Hz stimulation. So overall, this indicates that ongoing stimulation decreases the expression of protein kinase M-zeta. And we know that it's so important to have expression and function of protein kinase M-zeta for ongoing stability and maintenance of potentiated synaptic circuits. And let's wrap it up with a summary. From this lecture, I hope you can see that memory maintenance, indicated by behavioral measures, requires ongoing expression and activity of the memory protein kinase M-zeta, long beyond anything that we could measure in a hippocampal section in a bath as it's slowly degrading. The best evidence is that microinjection of the protein kinase M-zeta inhibitory peptide, or ZIP, or infusion of a viral vector encoding a gene that makes a protein that blocks protein kinase M-zeta, blocks enduring memory. And we saw similar effects with ZIP on LTP back in Chapter 7. It's also noteworthy that the actions of ZIP in blocking protein kinase M-zeta are transient because the ZIP is gone after 24 hours. Yet we see that this is sufficient to break down the ongoing function of ZIP to allow the spines to collapse back into their naive state, perhaps, and to have a breakdown in the maintenance of the memory process. I also want to remind you that overexpression of protein kinase M-zeta enhanced memory in behavioral tests, an exciting finding that may have potential for further investigation as we seek ways to deal with memory deficits in people with age-associated memory impairments and so on. But again, no directly relevant kind of clinical applications at this point in time. And finally, we saw in the realm of forgetting of memories that active decay theory is supported by evidence that ongoing activity, basal activity, or by moderate stimulation of calcium influx through NMDA receptors activates conventional protein kinase C isoforms that are not self-perpetuating and diminishes ongoing expression and activity of protein kinase M-zeta, resulting in loss of LTP and loss of memory maintenance in behavioral tests. So there we have it, the very important roles of protein kinase M-zeta that we saw back in Chapter 7 in relation to maintenance of LTP, and now that we see in the much longer term as being very important in terms of the maintenance of memory. And I'm going to stop there, so stay safe, and I will see you tomorrow with the lecture for Chapter 13.